Well, welcome everybody. We have a really special opportunity today to be talking to Dr. Hilary Gammill. Dr. Gammill is an associate professor at the University of Washington. She is also a physician scientist in maternal fetal medicine, so works at the Fred Hutchinson Cancer Research Center, and is the co-chair of the Preeclampsia Registry's Scientific Advisory Council. Busy lady. But this is a really special opportunity for us to talk to a researcher who has been very involved in a study that just came out. We're going to be talking about that study and kind of doing a deeper dive to understand the significance of this research and to really be able to highlight this particular study because it was truly enabled by the Preeclampsia Registry. And so this is a really cool opportunity to kind of unpack that and understand how that came to be, and I think especially like what is the significance of this research. So Dr. Gammill, just thank you so much for joining us today. Would you like me to call you Hilary or Dr. Gammill?
Yes, please call me Hilary. Thanks for the opportunity. This study analyzed the genetic data from 181 participants in the Preeclampsia Registry who had provided self-reported information about their experience with preeclampsia and had also provided medical records that were reviewed to confirm the diagnosis in that manner. And those 181 women provided saliva samples that, from which we extracted DNA and performed whole exome sequencing. And that data was compared to two reference populations, control populations, for differences in these 43 genes that have been found to be associated with cardiomyopathy.
So the other thing that we've known for a long time is not only is there some kind of a genetic component, like you said, it tends to run in families, not exclusively, because we know there are many people who don't apparently have any family history of preeclampsia, but there still is some kind of genetic component. The other thing that we've known about for a long time is that there is definitely some kind of a correlation between preeclampsia and heart disease or cardiovascular disease. Right. So that is something that is not emerging research. I mean, that's been pretty resoundingly supported for years in a number of different studies. The big question has seemed to be, is it that your experience with preeclampsia confer some kind of injury, increasing your chances of getting cardiovascular disease, so that's kind of a causation approach, versus a correlation approach, which is what predisposed you or what caused you to get preeclampsia the same thing that's going to cause you to get some kind of a heart disease or stroke. How does this study contribute to that question?
Right, and I think that question is so important, and it was the inspiration for this study. I think the answer in the end is probably going to be that it's both shared genetics and also conferred risk, but we really need to tease that apart. One of the inspirations for this study was a recent paper that was published looking at the relationship between unexplained cardiomyopathy, so a particular kind of heart disease, not associated with pregnancy, and peripartum cardiomyopathy, which is a particular kind of heart dysfunction that happens around the time of delivery. And they found some intriguing shared genetics within those two conditions. And we further hypothesized that those same changes would be associated with preeclampsia. And so we looked at the same set of genes in preeclampsia to see if there was sharing across preeclampsia as well as the other conditions. I think what we have learned is, again, pointing to the fact that there is heterogeneity in preeclampsia. As you said, not every preeclampsia is a familial preeclampsia. And some people are going to have some kinds of genetic changes and other people may have other kinds of genetic changes. But what we found with this study is that there is shared genetics across those three conditions. So there is, in some folks, an underlying genetic condition that predisposes to both heart disease, this particular kind of heart disease, and preeclampsia.
And tell us a little bit about the significance or I guess the weight of that correlation that was found? How significant is it? Or in this study?
In the study, there was definitely an increased representation of these genetic variants in the preeclampsia cohort that we studied, which was 181 women who were participants in the preeclampsia registry, compared with a couple of different control populations in general. And there was both an increased rate of those variants. There was also an increased number of variants per person. So some of the women with preeclampsia had multiple genetic changes that were associated. So there was, the findings were significant. I think it's really important in a study like this to caution that one of the difficulties always in science is reproducibility. And so really this needs to be verified in another population and confirmed before we can leap to actual clinical diagnostic implications. Yeah.
Can you help us interpret that? When we say that preeclampsia survivors had a much higher rate of these cardiomyopathy genes, what does that mean in reality? I mean, higher rate could mean slightly more. It could mean, what does it mean?
So I think, in terms of kind of translating it into a numerical impact, that's a little bit tough, I think. But I think what's important to take home is that there are a couple of different directions that the fields are going, and personalized medicine is a huge emerging field. And I think that if we start to understand some genetic changes that we can then potentially way down the line use to evaluate the type of genetic contributions that an individual has, then we can target therapy. And in a situation like this, you can imagine potentially intervening to prevent heart disease in women that we know are at higher risk for heart disease. And the genetic changes can help us know why and how to best do that. And in a larger context for the field, when you start to identify, especially reproducible, consistent findings, that identify genetic mutations, you can identify pathways that can be targetable for treatment and intervention. So you can understand maybe if this genetic mutation causes this problem, then you can do further studies to understand, well, maybe this is a better approach to understanding this type of preeclampsia, and how can we intervene to prevent that or treat that.
So you've used a couple of terms. Let's kind of unpack those for a second. First of all, the concept of heterogeneity. There is definitely a growing sort of mindset or framework around preeclampsia that is suggesting what we today just call preeclampsia is actually that there are many flavors of it potentially, or that the subgroups of people who get what we are today calling preeclampsia are actually pretty distinct kinds of people. And so, when Group A gets preeclampsia, they get it for a very different reason than Group B might get it.
Right.
So this concept of heterogeneity with this study can you kind of unpack that a little bit more?
You did that very well. I think that one really great example to illustrate the concept of heterogeneity when we're talking about, a mixed up population is diabetes. So if you consider there's type 1 diabetes and type 2 diabetes. And both of those conditions are related to high blood sugar. But how you get to the high blood sugar is completely different. In type 1 diabetes, you don't have enough insulin to keep your blood sugar under control. And in type 2 diabetes, you can't respond to insulin in the same way. It's completely different. One is sort of upstream and one is sort of downstream. And so how you get to the high blood sugar really matters in how you address the condition. And for a long time, the field of preeclampsia has kind of had a sense that there really are very different ways to get to the diagnostic criteria for preeclampsia. There are definitely different flavors or boxes, as you outlined. and really being able to look at an individual person and say, oh, you have this type or you fit in that box, is crucially important to being able to treat them well, and to intervene at the time with preeclampsia, but also to address downstream risk later in their life. And I think that what we've uncovered here with these genetic changes may help, especially with the later life consequences.
So to link that then, what we have found in this particular study is a fairly large group, and I want to, you said let's not get into the numericals, but I actually do want to get into that a little bit, but a fairly large group in comparison with the general population out there who have these genetic markers for cardiomyopathy and who had preeclampsia. So what could this mean? Let's unpack that in terms of what could this mean for preeclampsia, for sort of treating and intervening in preeclampsia in those childbearing years.
Sure.
And separately, what does this mean for the long-term prognosis for these preeclampsia survivors who also have these cardiomyopathy genes?
What these genes are associated with is cardiac dysfunction. So the heart muscle not working well. That's, it's a type of heart disease. It's not necessarily a cardiovascular disease, but cardiomyopathy, that condition means that the heart is not working well. And if these genetic changes are pointing to a pathway toward preeclampsia that's related to the heart not working well, then you can envision in kind of a trajectory longer term that you might be able to impact how the heart isn't working well and improve that with an intervention. So in the immediate term, you might be able to improve cardiac function and either treat preeclampsia or at least mitigate preeclampsia and extend pregnancy. In the longer term, since we know that women with preeclampsia are at later life risk of heart disease, we can potentially use the understanding of some of these genetic changes to know who and how to intervene to prevent that later heart disease or, again, mitigate the risk.
And that's where that precision medicine concept really comes into play. I want to get at some other interesting findings, because I know that in addition to this, one of the other things that was really interesting is the fact that so many of these were found in term deliveries or near term. Talk a little bit about that, because that seems very contrary to what we've been led to believe about the risk for CVD and preeclampsia.
Right, right. So as you were talking about kind of the heterogeneity of preeclampsia, the boxes that we've traditionally used to kind of understand where people fit are things like early onset, how early in pregnancy, and whether there are severe features. So whether there's evidence of organ systems and disease that is affecting more systems within the mom. And so typically when we look at things in those boxes, we know that the rate of cardiovascular disease, that association of preeclampsia with cardiovascular disease later, is higher in women who've had severe preeclampsia, severe features, or who have had early onset. So we know that that rate is significantly higher. It's about 8-fold compared to 2-fold higher in women with term mild preeclampsia. In this study, we were surprised to find that if you compared within the preeclampsia cohort, if you compared women who had one of these mutations or one of these variants with those that did not, there was an overrepresentation of term more mild disease in those that had a mutation. We don't have an explanation for that, but I think what's particularly intriguing about it is that it's hard in that term preeclampsia box that we talk about that have the 2-fold increased risk to know who to really focus on to follow to intervene for heart disease. And this could be a way to sort of stratify that group more to know who to really focus on, in addition to the traditional way of looking in particular at severe early onset preeclampsia.
So if I'm understanding this right, and tell me if this is not an accurate summary of it, we've traditionally said that women who had early onset or repeated cases of preeclampsia are the ones who are at highest risk for cardiovascular disease in their future. Sometimes not very far out future.
Right.
What this study is suggesting is that even those women who had preeclampsia at term, regardless of how significant it was, the fact that it was near term, would typically be maybe not dismissed, but just kind of put into this low risk category for future heart disease. And what this is suggesting is that that's probably not a really fair assumption, and that some of those women who have these genetic changes or variants, as you're saying, really should be looked at and followed more carefully and more closely, because they, even though they had term preeclampsia, are still at higher risk according to these genetic variants.
Right. I mean, this is the first time that we've found that, and it's
definitely early. Definitely would need to be validated through other studies, but it's also something that's quite different than anything we have seen in the literature to date.
Right. And it is not what we expected. We sort of expected that the boxes we've been using would be the same boxes, but I actually think in a way if it's reproducible, it could be more useful than if the boxes were as we expected them to be.
Sure. So let's talk a little bit about the preeclampsia registry. The registry was used for the study. Could the study have been done without the registry?
Absolutely not. One of the really important aspects of this study is that we were able to, first of all, it was amazing, the response from the participants and their willingness to provide saliva genetic samples for the study. We were able to get both their self-reported information and also copies of their medical records to confirm the diagnosis in all participants. And all of that information is now part of the registry, which will be shared and available as a resource to the research community. But the other really important component is that we were able to follow women up after their pregnancy, and even after we did the initial analysis on the study, we were able to get a large majority of the women to respond and provide an update on their health. And the ability to follow up long-term both maternal and offspring outcomes is so important, because it allows us to know the implications of these genetic changes, right? If we're wondering what the implications are of a cardiomyopathy related gene variant, then being able to be in touch every year to say, how are you doing, what's going on, gives us a real window into what that trajectory might look like and how it might differ. And it was amazing to me that even in the context of, while we were doing this analysis, we were able to recontact and get more than, I think it was 70%, it might have even been slightly more, participants to provide a new update was really astounding.
And that's something that I think is sometimes lost in understanding how something like the preeclampsia registry, how it differs from a traditional study,
right,
where you start off with a hypothesis, you enroll participants in that study, you collect data, you analyze it, and then you're done. And you really don't have the ability, either ethically or logistically, to stay in touch with those participants. And that's something that really is different about the registry. So the data that's been collected, the DNA that was collected, it went through whole exome sequencing, all of this has been gifted back into the registry. Right. So for any investigators who might be watching this video, what does that mean for them?
So both the data from the whole exome sequencing and the samples should whole genome sequencing or other sequencing technologies, other genomic technologies, be appropriate in the future. All of those things are resources that will allow the research community to investigate new hypotheses and adapt to new technologies, and also be able to collect that sort of phenotypic information, that what's happening over time to these women and their children, and to be able to look at both the both look at existing data to evaluate hypotheses, but also explore completely new studies. I think it's a really valuable resource for the field.
So it goes without saying that this is the first of its kind. This is the first time that these particular findings have been brought out and published and sort of put into the research community. Clearly, they'll need to be validated. Others will need to do similar studies and see if what we found was some weird aberration or something that's reproducible. I think one of the things from the patient's perspective is that patients are really keen. We have found so many people who are really keen to have their experience make a difference and contribute to research. Yep. And what the registry allows to happen is to maximize their contribution, because they're not just contributing to one study. This is data that's being used over and over and over again. And the fact that they can be recontacted has been really not only inspiring and helpful to investigators, but I think to patients who can realize, like, wow, I can really make a difference in what we understand about this issue.
Absolutely, and we are so grateful to every participant in the registry and in this study. And I think that there's a lot of power in kind of, I know it's an overused word, but almost crowdsourcing. So bringing patient involvement in, but really importantly, bringing the patient perspective in to inform new hypotheses, which I think is really key. And also then allowing the data to be crowdsourced to investigators of all sorts of different backgrounds and perspectives to really try to answer things. I think that there's a lot of value in open sourcing of all of this information so we can move the field faster.
Thank you so much for your time, Dr. Hilary Gammill with the University of Washington and Fred Hutchinson Cancer Research Center. This has been a deeper dive into cardiomyopathy and preeclampsia. Shared genetics? We think so. Thanks so much.
Thanks, Eleni.
Bye-bye.
Bye.